Good afternoon, everyone. My name is Sarah Busch. I'm the Chief Scientific Officer of Healios NA, and today I'll be presenting to you primarily on our global phase 3 trial for treatment of acute respiratory distress syndrome, or REVIVE ARDS. Yes, I need this. So Healios, for those of you who don't know, is Japan's leading cell therapy and regenerative medicine company. Healios KK is the parent organization. It's been in this space for over 14 years, and they've recently acquired the Invimestrocel or multi-stem cell therapy platform, which we are now advancing to a global phase 3 trial in ARDS. This trial is built on a foundation of multiple regional phase 2 clinical trials in ARDS. And we're now able to, given our positioning in Japan, leverage a very favorable regulatory framework built by PMDA and on track to achieve a near-term conditional and time-limited approval for product launch in Japan. We've been granted orphan designation in ARDS in Japan, as well as fast track and RMAT with FDA in the United States, which positions us well for an accelerated global development path. We have a core strength at Healios in manufacturing, which, as many of you know, is really critical, and we see this as a key competitive advantage. Our leadership team has broad strengths in both the science and business of regenerative medicine, which has poised us, again, for being a global leader in cell therapy. This is our regenerative medicine pipeline, and Healios has been a pioneer, as I said, in the cell therapy space, starting off working in replacement cell therapies with an RPE technology many years ago, which has now advanced to partnership with Sumitomo Pharma. Healios has deep expertise in iPSC biology, which led them to develop NK immuno-oncology therapies, which have now partnered with Akatsuki Therapeutics for advancement. But as you can see, our most advanced areas of our pipeline are based on the Invimestrocel or multi-stem cell therapy. And we have the ARDS program that I'm going to focus on today, as well as programs in acute ischemic stroke and in trauma, which is a study ongoing at a single site at UT Houston. So what is Invimestrocel? It is a bone marrow-derived adult cell therapy. So this is an allogeneic product which requires no tissue matching. It's not modified, no genetic modification, no additional steps. So it is on-site, prepared for IV administration. Doses up to 900 million cells per dose in the ARDS indication. So we're very well positioned for our work in acute critical care because of this vialized product that can be stored, again, on site, which then simply needs to be thawed, cells removed with a syringe, injected into an IV bag, and then administered directly to the patient. So because time is so critical in these patients, we believe that we can get them there within 1 hour of an origin point from the pharmacy to actually treatment at the patient bedside, so very, very rapidly. Invimestrocel is distinct from other cells like mesenchymal stem cells that are quite well known in our space through a number of characteristics. So they have very robust expansion capabilities. Their technology is based on the MAPC technology, which you may be familiar with, which enables them to expand via a proprietary process, enabling expansion up to very high population doublings and allowing us to manufacture potentially hundreds of thousands of doses of product from one single bone marrow donation. So really robust expansion, and this all enables us to have a high-quality, reproducible product that can be, again, given to many, many patients. The cells are phenotypically distinct from MSCs, as I said, which we continue to measure on our manufactured product, and they're a smaller size, which actually impacts their biodistribution, and I'll show you that data in a bit. Invimestrocel is a living and dynamic cell product, so it's actually capable of interacting with the system, unlike something like an exosome product or a conditioned media product. This is a living cell product.
And we know over decades of experience and research with these cells, I couldn't fit all the publications on here, so it would get a little bit too wordy, but they interact with multiple different cells of the immune system. But we have narrowed down the mechanism of action and developed supporting potency assays and assessments, characterizations that we run on our batches of material that allow us to ensure that we have an active and potent product for treatment of patients. At Healios, we believe we have the most advanced allogeneic cell therapy bioreactor platform in the world in this space. Like many others, we started working in 2D cell factories, and this was where we produced our initial product, but now we have moved to single-use bioreactors and completed multiple production runs and intend to move forward to commercialization with a 50-liter bioreactor process. We've recently been granted a $47 million grant from the Ministry of Economy, Trade, and Industry, which is a subsidy to support our manufacturing scale-up. And we have already, in process development, moved through 200-liter and up to 500-liter bioreactors, which, again, we believe to be relatively unprecedented in this space. And this will enable us to lower our cost of goods and to generate sufficient product to treat a global patient population for multiple indications. So the ARDS indication itself is a very devastating disease. ARDS is an inflammation-mediated acute inflammatory disease that occurs very suddenly in critically ill patients. It impacts over 400,000 people each year in just the US, EU, and Japan alone. It has incredibly high mortality rates, ranging from 30 to 58%, depending on source geography. And there are currently no approved products. So mainly standard of care is treatment of the underlying cause, for example, with antibiotics with a bacterial pneumonia. But currently only respiratory support, including ECMO, are indicated in the management of moderate to severe ARDS. So lung protective strategies are used typically in the ICU in management of these patients, but invasive mechanical ventilation itself is known to be associated with very negative long-term outcomes. And we've talked to many patients who have lived through ARDS and have had very devastating long-term complications as a result of the treatment of IMV itself. And would not wish that on their worst enemy. So our goal is to get patients off of these ventilators as soon as possible and reduce all of these complications that go along with being on IMV. ECMO, as I mentioned, is a potential strategy, but its availability may be limited, and it can only be applied to a certain patient subset. Getting down into more of the cellular level of what goes on in ARDS that can be impacted by Invimestrocel, you can see that there's an initial clinical insult that may be due to trauma or an infection like pneumonia. And again, we've restricted ourselves primarily to looking at pneumonia-induced ARDS. And so what happens is there's a pro-inflammatory response of the cells in the lung, which creates a feedback mechanism. So more pro-inflammatory cytokines cause more recruitment of pro-inflammatory cells and so on, which leads to hypoxia developing in the lung and the patient falling into severe respiratory failure. When we treat with Invimestrocel, the cells naturally home to the lungs as a result of injection IV through the pulmonary circulation. So they're exactly where they need to be at the time that they need to be there. The cells then interact with these immune cells in the lung and suppress this inflammation and suppress the ongoing course of the disease, allowing for it to resolve. And the hypothesis is that this will continue to lead to earlier ventilator removal and a reduction in mortality. And this hypothesis has been well proven out in non-clinical animal models. Again, we have multiple publications that support all of this data, demonstrating a reduction in inflammation, demonstrating repair of the injured lung tissue. And I'm just showing one example here of human ex vivo lungs that were rejected for transplant, but clearly you can see there's a significant amount of inflammation ongoing in the panel on the left-hand side here. And then when the lung is treated with Invimestrocel, the inflammation was significantly reduced. Probably hard to see here. This is an inflammatory score done by an unbiased pathologist. And we also saw a reduction in pro-inflammatory cells, including macrophages primarily, and neutrophils, which are two key cellular players in the progression of ARDS. We've also shown that pro-inflammatory biomarkers are reduced both in ARDS and across multiple other non-clinical indications, and we have progressed to measure these biomarkers in the clinical setting, and I'll show you that as well. I mentioned we've conducted 2 phase 2 clinical trials to date in ARDS, both focusing on moderate to severe ARDS. The MUST-ARDS study was conducted in the US and UK and enrolled 36 subjects. And the OneBridge study was conducted in Japan and enrolled 35 subjects. And I will walk through a little bit of the data for you, so I don't have to go through our conclusions yet. MUST-ARDS was primarily designed to assess safety, and I know I have it in a footnote here, but I want to emphasize that we saw that the product was well tolerated throughout the study with no severe adverse reactions related to the product. And the study was randomized 2-to-1 for treatment to placebo, and we saw a substantial reduction in ventilator-free days and a trending, but we believe clinically meaningful impact on mortality. This observation was maintained when we looked at more severely sick patients, so those that had a low pulmonary function. We still saw a substantial reduction in ventilator-free days and improvement in mortality. And this always brings up the question, are we keeping patients alive in the short term and saving them from their acute illness only to have them suffer potentially or live low quality of lives moving forward? But in fact, when we looked at quality of life, over a year, at every time point measured, the Invimestrocel-treated patients did better than those placebo-treated patients. And in fact, at 1 year, twice the number of subjects that were treated with Invimestrocel rated themselves as completely independent in self-care. And I think this is very, very impactful. The OneBridge study largely recapitulated these results. It was a 2-to-1 randomized cell therapy to placebo. And we saw, again, a substantial reduction in ventilator-free days and a trending reduction in mortality that was quite substantial. 2 minutes left. I'll go through this data quite quickly, but there was another pre-specified analysis in this trial which allowed us to compare our data with matched historical data that had been generated in Japan in the ARDS indication. And so this was a 1-to-1 randomized with the Invimestrocel-treated patients to the matched historical data, and it largely, again, recapitulated the findings that we saw in the actual randomized trial. We, of course, being scientists, had to go ahead and pool the data from these 2 very similar trials that seemingly had very similar outcomes. So this gave us 60 ARDS patients to compare to 20 placebo or standard of care. And again, this was a mostly pneumonia-induced ARDS cohort of 85% Invimestrocel-treated patients and 90% placebo having pneumonia as their primary cause. And again, this was 900 million cells delivered at day 3 to day 4 after diagnosis. And again, our findings of the post-hoc data recapitulated our observations regarding ventilator-free days, making a very substantial impact in reducing these days, both across mean and median. And this is a very important piece of data. So I mentioned that in the MUST-ARDS and OneBridge studies, we looked at 3 to 4 days post-ARDS diagnosis as a time for intervention. And when we map this out and look at it graphically, we can see that as time to treatment reduced, we actually saw a greater benefit. And for that reason, we have focused in on treating in less than 48 hours for the current phase 3 clinical study. And as you can see, this was superior across the ventilator-free days and gave us greater ventilator-free days. I mentioned that we had looked at acute inflammatory biomarkers in a number of non-clinical studies to date, and we did so as well within the MUST-ARDS trial, and we saw a significant reduction in pro-inflammatory biomarkers in Invimestrocel-treated patients. And I don't have the data to show you here. It's a bit complicated to look at, but across all the biomarkers, as we look over time, you can see that they continue to reduce as you reduce time to treatment. So that brings us to our global phase 3 study for pneumonia-induced ARDS, which we plan to launch imminently. And this study will, again, look at a 900 million cell dose within 48 hours of meeting that ARDS diagnostic criteria, as I showed you earlier, seems to be better. And the patients will be measured as a primary endpoint on ventilator-free days at ventilator-free day score, at 28 days, and this endpoint has been agreed to by FDA and PMDA regulators. We have interim analyses built into this adaptive trial design in order to look and ensure that we win in the study. And so our initial look at efficacy will be at 300 patients, which is where, if we recapitulate the data that you saw to date in the 2 phase 2 clinical trials, then we would achieve success at that 300 patient endpoint. However, we have potential to enroll up to 550 patients if not stopped before that point. So as you can see, we have a lot of work to do ahead of us, but our 2 primary goals are to launch Revive ARDS, which will actually begin in Japan, and then expand that trial to US and rest of the world, and of course to obtain that conditional and time-limited approval and move towards product launch in Japan. So it's a very, very exciting time. I've been working on this therapy for over 14 years now, and to be on the precipice of an approval in which I think we can make a really meaningful impact for patients with ARDS is really exciting. So thank you so much, and I'll be here. Richard Kincaid is here as well if there's any questions as follow-up. Thank you.